Good morning, everybody, and welcome to the day 2 of our workshop. Very happy to welcome Hans-Peter Kiem this morning, who's going to give our first talk in our gene therapy cure research in animal model session. Before I hand the podium over to him, though, I'd just like to let the speakers know that today we have some special cards, which perhaps they were there yesterday, but I did not see them. Cards that allow me or the session chair to show you how your time is going. Okay, everybody get the idea. So you'll be seeing these cards potentially. Anyway, thanks very much, Hans-Peter, and I'll look forward to your talk.
Well, thank you very much for inviting me. It's a pleasure to be here and speak today. Let me start with this slide, of course. I probably don't have to explain much, but of course, Timothy is certainly was the first one cured of HIV. And we also know that compared to the Boston patients, Timothy stayed cured, even though the Boston patients, is there a pointer, by the way? In contrast to the Boston patients, he stayed in remission, has still been without HIV now for more than 12 years, although the Boston patient also had a somewhat prolonged remission from 12 to 32 weeks after their ART removal. But clearly the difference I want to already point out, they both had the allogeneic transplant, they both had conditioning. The difference was, of course, the CCR5 delta 32 versus the wild type CCR5. Now, this has recently been repeated, as you know, certainly one more patient in London, probably also the Düsseldorf patient, and this was an article in the New Yorker here, as you can see, where both of these cases provide a proof of principle about the potential of stem cells that lack CCR5. So I've been really thinking in terms of cure. We've certainly been focusing on two aspects. Cell protection, this can of course be done either with CCR5, the CCR5 delta 32, or some anti-HIV activity, of course, in the allogeneic setting. This is graft versus host or graft versus HIV reservoir. But again, those are the two things we've been focusing on in Seattle. That's the cell protection and then also providing the cells with some anti-HIV activity. So we've been modeling really this in non-human primates, and we've been focusing on stem cells, hematopoietic stem cells, for a simple reason, as you can see. If you put a particular gene into stem cells, it will be in all the different lineages here. Of course, for some diseases, it's important to have the gene in red blood cells or expressed in red blood cells and white blood cells. But really, for our purposes, as you know, it's important to have the T cells and the immune system protected. And then, of course, also the monocyte and macrophages. And again, this can all be accomplished really with stem cells, with the modification of hematopoietic stem cells. This just shows you, again, what we can do with hematopoietic stem cells. We can, of course, again, put CAR constructs here into stem cells, and they will also be expressed in the lymphocytes. We can also put, as you can see in the next slide here, broadly neutralizing antibodies or ECD4IG constructs in there. So these stem cell-derived cells can not only make CAR T cells, but also express broadly neutralizing antibodies or these ECD4IG molecule. And then, of course, we can also protect these cells either with CCR5 or virus fusion inhibitor. And we've been working with the C46 there, as you will see in the next few slides. One important aspect I mentioned before is, again, the macrophages, and this was a report that came out earlier. We've been tracking, of course, our animals to see where these genes, T-modified cells, go, and this just shows you very nicely after a stem cell transplant in the non-human primate, these cells traffic very nicely into the brain as well. This just shows you GFP cells and then your microglial cells. The yellow is sort of double stained. The red is the IBA microglial stain. So we could very nicely show that these stem cell-derived cells traffic also to the brain, so really to the reservoirs of HIV. So we've been focusing on two mechanisms to protect the cells from HIV infection. One was the C46 fusion inhibitor, and the other one, the CCR5 disruption. We've been using different types of nucleases, initially the zinc finger nucleases and more recently the CRISPR-Cas system. So with the C46-based system, we've done our initial study now a few years back. This just shows you a control vector with no C46, no fusion inhibitor, and then over here we've done monkeys with the C46 fusion inhibitor. We put also in a selection cassette so we could actually increase the number or percentage of gene-modified cells in the animals. So we modified these cells, transfused them into the animals. Again, this is without C46, with C46, and then selected them so we had about 20 to 30% gene-modified cells in the animals, and then challenged them in vivo with SHIV. And these are the results. Again, we've published this a few years back now with 89.6 P, but you can very nicely see the controls, they succumbed to AIDS, and the animals with C46 transduction very nicely recovered their T cells and actually remained without any symptoms of AIDS. In a separate set of animals using actually a clinical grade vector with CalImmune, also but using the same C46 fusion inhibitor, we follow these animals much longer, and as you can see, even over time, the viral load went down to almost undetectable levels. So clearly those animals were protected long term. They had virus-specific T cells, but the virus was not completely gone, but at that point you can see 800 days after transplant. That's a fairly substantial time without any development of AIDS in the animals. This is the control without gene-modified cells that was published by Schulak, again, quite a few years back. So the next step was, of course, now looking into CCR5 modification here, because this was easy to do with gene editing. This previous study was done with gene therapy using a lentiviral vector. The CCR5 modification is done with gene editing. Here, as I mentioned, we've initially started with zinc finger nucleases. And since we switched to the CCR5 targeting, we also had to switch to an R5 tropic SHIV virus. And here we've been using the SHIV 1157 IPD 3 and 4 from Ruth Ruprecht, which is an R5 tropic SHIV. Again, the envelope here is from the H1 clade C. We've set up the system so we could infect the animals and then suppress with antiviral therapy. Down here is the regimen that we've been using more recently. And this has actually worked very nicely. So we can infect the animals. You can see we get very nice set points for about 6 months here. We can then, down here, suppress the animals with this drug regimen that I just showed you. And then it was actually very nice to see pretty much all animals had rebound after we took off ARD, which is very nice and confirmed that this is actually a very robust model for our studies. So in many of our studies, we've been looking at different interventions to prevent the rebound back here. So that confirmed that we could use this as a model to study these different interventions. So as I mentioned, next we went initially to zinc finger editing of the CCR5. And again, a lot of this work was done really spearheaded by Chris Peterson in the group. Very straightforward. We take CD34 cells from the animal, bring them to the laboratory, and electroporate them overnight with these zinc finger mRNAs to knock out CCR5, and then reinfuse them back into the animals after conditioning. The efficiency ex vivo with the CD34 CCR5 modification was actually quite efficient initially, as you can see, about 40 to 60%. But then when we went in vivo, you could see it dropped down to about 5% or even lower in some animals. But the encouraging part was the modification was multi-lineage. So we clearly saw it in B cells, monocytes, and T cells. So clearly we were able to modify stem cells and multi-lineage cells in these animals. The encouraging part was also, even though the efficiency CCR5 modification was low, we saw a very nice selection of these especially the effector memory T cells here, and the terminally differentiated effector memory T cells here. This is fold increase after we then infected these animals with the SHIV-C virus that I just showed you earlier. So this looked very encouraging to us. but of course the efficiency was quite low. What was also encouraging, a recent publication we've had, is that when we transplant animals with delta CCR5, so with stem cells that were treated with these zinc fingers to knock out CCR5, we actually saw less tissue-based shift DNA and RNA in these animals compared to our control animals. So clearly there was an impact even in the reservoir when we transplanted with a delta CCR5. Now, in terms of efficiency, I just showed you it was fairly low with the zinc fingers, but more recently we had switched over to CRISPR-edited cells. And this is to show you from a different study using fetal hemoglobin reactivation, where we've actually shown now very efficient gene modification, at least now this is by Macaques here in the 30 to 40% range even now. So this is going out fairly long time, as you can see. So I think with CRISPR nucleases, we can substantially increase the overall gene editing efficiency. And this is what we're now also proposing, of course, for the HIV or HIV studies in our animals. So now I'd like to move on to talk a little bit about some work we've done in collaboration with the Kitchen and Jerome Zach groups. As you probably know, as I mentioned earlier, we can put CAR T cells, the CAR construct, into the stem cells, so would be expressed in the T cells and, of course, also NK cells. And we've just recently published this now a couple of years back, almost in PLOS Pathogens, as you can see here. So the idea here was we take the CD34 cells again, modify the CD34 cells with a CAR-containing construct. And again, these are constructs from Jerry Zach and Scott Kitchen, where we have a CD4 CAR with and without the CD3 zeta, so one that is actually active and one that serves as a control, shown down here. This recognizes the GP120. So we could really see can these cells engraft, and how long would these CAR T cells then, stem cell-derived CAR T cells, contribute to repopulation in these animals. And this is just shown here, the overall gene transfer efficiency. This was using a lentiviral vector to introduce the CAR construct. And again, the CAR construct also had a C46 that I've just shown you earlier to protect the cells. So they're really defensive protection, plus, as I mentioned earlier, this anti-HIV activity here in form of a CAR construct. It was very nice to see we could use a human CD4 to really track these CAR T cells, because it didn't cross-react with the monkey CD4. We saw very nice engraftment, though low levels of these CAR-modified cells, T cells. But it was very nice to see we saw CAR positive cells in the T cells, in the NK cells, and in the B cells, of course. So really very nice that we can get the CAR construct not only in the T cells, but also in NK cells. And most of you know it's quite, CAR NK cells have become also a very important way of killing cancer. And we hope, of course, this will also be relevant for HIV. Now, this was very nice to see how these CAR constructs really functioned in vivo in the animals, because we could track these effector cells. I just want to show you, this is the baseline before we infected the animals. When you just look at the memory is blue, gray is naive, and then the effector. This is before infection here. When you then look at infection, you can see how nicely these effector T cells go up. So there was a clear response. When we put the animals on ART, this went down again. And then when we took the animals off ART again, this went up again. So clearly they were functioning in vivo, these stem cell-derived CAR T cells, after transplantation and then also repopulation in the animal. Now, of course, since we've done this in a non-human primate model, we could really screen all the different tissues where these CAR T cells went. Isaac Barber, ex-Bell in the lab, he did his master thesis in the lab, was using immunohistochemistry to really look and track these CAR T cells, stem cell-derived CAR T cells. And shown here is that they traffic very nicely to the lymph node germinal centers. But also, again, very important, of course, they traffic very nicely into the brain. And here to show again the merged, down here, the merged ones, the IBAs, again, the microglial cells are red, Of course, the green cells are the GFP-positive cells. Then you can see some merged cells with the IBA, the microglial cells, and the GFP. So clearly these CAR T cells, stem cell-derived CAR cells, also migrate into the brain and hopefully can eliminate HIV in the brain, any reservoirs in the brain. So I think with stem cell-derived CARs, we can overcome some of the limitations of T-cell-based CAR T-cell therapy. The stem cell-based CARs persisted for more than 2 years, and again, were actually active when we took the animal off ART. They responded again very nicely. The stem cell CARs may again correspond and wax and wane depending on the viral antigen presence in the animals. And then they also very efficiently traffic to B cell follicles and other potential reservoirs. I didn't show you. We also looked, of course, in the GI tract, and I've shown you the CNS. So very nicely, again, I think stem cell-derived CARs could traffic to the different sites. Now, of course, we can not only put these CAR constructs into stem cells, we can also now put broadly neutralizing antibodies, and we've published this also just a while back now. And then we can put, of course, these ECD4 Ig molecules in there as well. So we can really have two activities in there in these stem cell-derived CARs. We can, in these stem cell-derived cells, not only the CAR T cells, but also, again, B cells, B broadly neutralizing antibodies, or these ECD4 Ig molecules to treat HIV and hopefully prevent reactivation of HIV and eradicate HIV. This is just to show you the efficiencies overall keep increasing, I have to say. The efficiency of gene editing, even knocking in at particular sites. This is just using SSODNs to mediate HDR homology-derived recombination. And we could get up to 80% overall looking at indels and insertions. So that efficiency is actually increasing quite dramatically. And of course, we're also working on different ways of conditioning the animals. So we've been working quite a bit with Magenta as well to utilize non-genotoxic conditioning with either CD117 or CD45, so we could avoid any potential genotoxicity. So this could really be applied to patients with HIV, and not only with patients with HIV and underlying cancer. And that was it. I think my time is up. Thank you very much. I'd like to appreciate, of course, a large group of people that really have made this possible. Thank you.
Thank you. We have a couple of minutes for questions. Hans-Peter. So could you comment, you did to some degree, but what do you think is ultimately going to increase the ability to enhance the efficiency of these processes?
What I've not gotten into, we think, of course, we focus on a more subpopulation, like a CD90 population, where we see higher efficiency of editing. So it's it's only about 5% to 10% of the overall CD34 cells. We get very high efficiency in that highly enriched stem cell population. And then really figuring out to get cells into cycles, stop them again with cycling. So there's many different ways, but I think we're overall increasing with the editing efficiency. That's why I just wanted to show you. And we're never going to get 100%, but I do think we get quite substantial editing efficiency now, and most people would claim up to 70, 80%.
Considerations that one must consider when thinking about which types of animals are eligible for hematopoietic stem cell transplantation. DMV, herpes B, things that some people think about as SDF. I mean, SDF means specific pathogen-free, but you know that they don't have the things you thought about.
Not really. The things we've really been thinking about now mostly is when we use lentiviral vectors, we've been using the pigtails for editing the rhesus monkeys. Those are all considerations.
So you've successfully transplanted an animal that had DMV and or herpes B.
not herpes, we're screening for herpes, but CMV we can treat, yes. No, so we see CMV reactivation, but we can treat this really like in patients. Cytophagy works quite well, actually.
So these super clean monkeys that people are talking about, you don't necessarily think are super important for doing?
Well, no, that would be great. Of course, if we don't have CMV, we don't have to give any additional drugs. No, that would be wonderful. No, absolutely.